Hello everyone, welcome to one more video of special BIO-TONIC. Students, in our last lecture we talked about structure of DNA. In this session we'll talk about structure of RNA in detail. So watch the video till the end. Students, we know that RNA is made up of ribonucleotides. These ribonucleotides are in turn made up of three components: nitrogen bases, sugar and phosphate. We also know that nitrogen bases are of two types, purines and pyrimidines. Purines are of two types, adenine and guanine. Pyrimidines in case of RNA are cytosine and uracil. Students, in case of RNA, adenine pairs with uracil, whereas guanine pairs with cytosine. The sugar present in the RNA is ribose sugar, which is C5H10O5, and the phosphate is same, that is H3PO4. Students, this is the structure of ribose sugar, which is C5H10O5. This is carbon number 1, 2, 3, 4 and carbon number 5. Now, 1 dash is called 1 prime. So this is 1 prime carbon, 2 prime carbon, 3 prime, 4 prime and 5 prime. So students, the central part of a nucleotide is made up of ribose sugar in case of RNA. Now, when we attach base to the sugar, it is called nucleoside. So, a nucleoside is base plus sugar. Students, when we attach phosphate group to the 5 prime carbon, it becomes base plus sugar plus phosphate. It is now called nucleotide. So, a nucleotide is made up of base plus sugar plus phosphate, which is equated to nucleoside plus phosphate. So this is the structure of ribonucleotide. So we can define RNA as a polymer of ribonucleotides. Now students, to understand structure of RNA, let us see the structures of these nucleotides. Now the first nucleotide is adenosine monophosphate. Second is cytidine monophosphate. Third is uridine monophosphate. And fourth is the guanosine monophosphate. Now, the second nucleotide, that is cytidine monophosphate, is attached to the first one by using phosphodiester bond. Now, these are a covalent bond which are not easy to break. Now, the third nucleotide is added to the second one by using this phosphodiester bond. Fourth nucleotide is attached to third one by using this phosphodiester bond. So students, the RNA is made up of ribonucleotides which are joined together by phosphodiester bond. Now normally the RNA is single stranded, but part of it can become double stranded due to complementary base pairing. The direction of this strand is 5 prime to 3 prime. Students, there are two types of RNA. One is non-genomic RNA and second is genomic RNA. In case of non-genomic RNA, it doesn't act as genetic material, whereas genomic RNA acts as genetic material in case of retroviruses. This non-genomic RNA is divided into two types, non-enzymatic and enzymatic. Non-enzymatic are of three types: messenger RNA, that is mRNA, ribosomal RNA, that is rRNA, and tRNA stands for transfer RNA. Sometimes RNA can also act as enzyme. The good example of it is ribozyme. And third is the genomic RNA. The examples are retroviruses. So students, to summarize, RNA is of two types, non-genomic and genomic. Non-genomic can be non-enzymatic, which are mRNA, rRNA and tRNA. Enzymatic can be ribozyme. In case of some viruses which are called retroviruses, it acts as genetic material. Students, let us talk about the differences between the non-enzymatic RNA. Now, these non-enzymatic RNA are of three types: rRNA, mRNA and tRNA. Now, rRNA stands for ribosomal RNA, second is messenger RNA, and third is transfer RNA, which is also called soluble RNA. Now, rRNA constitute 70 to 80% of total RNA. mRNA is around 3 to 5%, whereas tRNA is 15 to 20%. Now, when RNA is produced during transcription process, which we are going to learn in the 12th standard chapter, that is molecular basis of inheritance. Now, these RNAs are produced by some enzymes. These enzymes are called RNA polymerases. Now, these three different types of RNA are produced by three different types of RNA polymerases. So, RNA polymerase 1 produces rRNA. mRNA is produced by RNA polymerase 2, whereas tRNA is produced by RNA polymerase 3. Students, in case of rRNA, it combines with proteins to form ribosomes, and we know that these ribosomes are the factory of protein synthesis, which help in the process of joining amino acid to form the proteins as such. So RNA combines with proteins to form ribosomes, which are factory of protein synthesis. mRNA act as the messenger to carry the message from DNA to cytoplasm for protein synthesis. So students, mRNA carries message for protein synthesis. Now we know that during protein synthesis we require various types of amino acid. These amino acid are brought from cytoplasm to the site of protein synthesis by various types of tRNA. So the job of the tRNA is like a trucks, which are nothing but goods carrier. So these tRNA carries the amino acid from cytoplasm to the site of protein synthesis. Now, as far as structure is concerned, rRNA has a complex structure. mRNA has a linear structure. If you look at this diagram, it has a start codon. The middle region is called coding region, and it ends with stop codon. So protein synthesis begins at start codon, and the set of protein synthesizing machinery reads the coding region and it stops at stop codon. So start codon and stop codon are the signals for beginning and end respectively, whereas the coding region is the one which is actually coded to form proteins. Students, for the time being, don't consider the words like exon and introns. We'll talk about exon and intron, such words in detail in the concept of protein synthesis next year. Now the tRNA has the cloverleaf-like structure or hairpin loop-like structure. Now let us see the structure of tRNA with the next slide. Students, this is the structure of tRNA. Now if you look at the structure carefully, on the left side it has D loop, on the right side it has T loop. On one side towards 3' end, it is attached to amino acid. The end of the tRNA which is attached to the mRNA is called anticodon. Students, this tRNA shows various structures such as D loop, T loop, anticodon loop, and the fourth place is where amino acid is attached. Now this tRNA brings various amino acids from cytoplasm to the site of protein synthesis. So students, I hope you're clear with the structure of RNA and its types and functions. So stay home, stay safe. Thank you.